see. Alrighty, so I think it's recording. Let me see. Alrighty, so since I think we have a virtual audience today, I'm gonna hear my talk, my intro to the computer. So hello everybody. Welcome to the final colloquium session of the year. I am so excited that we are here, but I'm also kind of bittersweet that we're at the end. But we definitely have saved one of our best speakers for last, so I'm really excited about this. So with that, I'm going to go ahead and introduce our guest speaker. This is Dr. Michelle Jensen, who is an instructor in the Department of Biology here at UWF. Dr. Jensen holds both her Bachelor of Science and her PhD in Biology from Louisiana State University, where she built a strong foundation in molecular biology and genetics. Her doctoral research focused on microbial antibiotic resistance, and she later continued her scientific training as a postdoctoral researcher at the Pennington Biomedical Research Center. There, she investigated the molecular mechanisms behind genetic obesity. This work that has been published, good grief. This work that has been published in respected journals such as Obesity Research and the FASE. Hope I said that right. At UWF, Dr. Jensen teaches a wide range of undergraduate courses, including molecular biology, general biology for majors, forensic biology, and several laboratory-based classes. She is an enthusiastic mentor, guiding both undergraduate and graduate students through directed study projects that explore cutting-edge topics like forensic DNA analysis of degraded samples and the use of the human microbiome in forensic identification. Please join me in welcoming Dr. Michelle Jensen.
Thank you very much. I hope you're all doing well. I'm very excited to be included in the program. I used to be a forensic DNA analyst, but I was lucky enough to now be able to teach classes and incorporate some of that. So I'd like to talk a little bit about forensic DNA analysis today without going into too many extreme details. And towards the end, I'll give you some interesting cases to kind of draw it all together. When we think of forensic DNA analysis, we're like, okay, here's a crime. And where's the DNA evidence that shows who did it? And so in one example of a DNA profile, you might have something that looks like this, where you see the word suspect 1, 2, and evidence. But what are those numbers? What are the blue peaks? Where are you getting that information? Where are they found? But a typical data printout for a file would be that. For a criminal case, we'll talk about what they are in a bit. So forensic DNA analysis has a lot of different applications. Just a few we'll talk about, but the main part will be just how do we get those peaks, those numbered, the data that we call the DNA profile. But don't worry, I'm not going to go through too much of this, but we'll get there. A couple of interesting cases, and then obviously I've got my references there. Paternity testing, you're familiar with that. The same type of DNA analysis can be used or is used. And when I say the same type, generally you can purchase reagents in a form of a kit for the specific goal you want to use. So if we're just doing a paternity test, right, the kit reagents would be specific to that. But there are different laws or different research regulations or guidelines to follow for different situations. And so, as you would imagine, for criminal investigations, that is much more tightly controlled and regulated. I'm not going to go into the laws about that, but just know that when you're doing those types of DNA analysis, you have to make sure that you follow every bit of the FBI quality control standards and so forth. So I've never done any of these down here, but you can imagine using these techniques for 9/11. Whenever paleontologists find new bones or researchers find new samples, it doesn't have to be a bone. It could be a piece of pottery, and maybe you're trying to see if there's plant material or whatever. So beyond the human DNA analysis, you can look at other species. You can look at other things. And then some of the more current ways to use a DNA profile or these techniques would be in agriculture or industry, and I'm not going to touch on those. What I tend to follow along with would be the criminal investigations. So we're just look at the sources of DNA. There are some that are better than others. In other words, if a CSI goes to a crime scene, they have been trained to know, first of all, how not to contaminate a crime scene, but how to observe, find applicable samples, how to collect those, following all chain of custody laws, packaging, labeling, all of those things that you would be accountable for. Regions of interest goes back to that idea of where are we finding those peaks? Where do those peaks and those numbers come from? Well, parts of your DNA are being detected, and we will talk about that and briefly mention these as far as how those parts of your DNA are detected. So what are the typical sources? And I'll mention why I've got some of those crossed off. Blood, semen, hair, and then of course, tissue and other types of cells. Those are going to be the body fluids or cellular material that will hold most of your DNA, right? So if you just looked at red blood cells, they don't have DNA in them. Other types of blood cells do. These here are much more difficult because of the chemical nature of those. So when FDLE comes to give their presentation, they always say, what's current as far as testing? And so that's why I just crossed those out, is because their latest or one of their latest presentations mentioned that they're less and less and less testing for these things because what they have found is it's very, very difficult to get enough data from that. So these types of biological material have to have nucleated cells because the DNA has to be there. And they're often found on the ground, blood on the ground, whatever it is. But you could probably come up with those scenarios thinking about some of the things the CSI might test looking for some of these things. So the word touch DNA, oh, you've handled something, really that goes back to touching it, your epithelial cells, your tissue, and the rest would be pretty obvious. Some of these are much better than others. So again, CSIs would be very well trained on what to pick up, what to test, and so forth. So again, just mentioning for blood, it's not your red blood cells. There's no DNA there, but the other nucleated blood cells. For semen, it's not the fluid, it would be the sperm. It drives me crazy when you see things on TV and some of these shows that say, oh, use the hairbrush and that'll be fine. Well, maybe if the hairbrush contains hair with skin cells or epithelial cells, and I'm not sure how well you can see that, but if you compare it from here to here, so this would be a pulled hair, and if you were to pull one of your own, you'd see the little whitish material. Sometimes it's kind of ragged looking, sometimes it might be more well-formed, but if you have a shed hair, it's falling out by itself, it's not carrying the tissue with it. So this will give you nothing in the way of your DNA that we're going to need to test. All right, so regions of interest. What the heck are those blue peaks that we first saw, and why are we putting numbers with them? Populations were analyzed. So we have Caucasian, Hispanic, different areas of the Hispanic populations, Black, different island populations, and so forth. Volunteers from each of those populations either provided a blood sample or some DNA sample. Scientists went through and tested, okay, what regions are going to be good for us? What can we use to build these DNA profiles? Because we want something that is unique for a person, like their fingerprint. We want something that does discriminate them, does have them stand out from everybody else. So when you think about these regions of interest, it took a long time, large numbers, maybe 300 per this population and so forth, a lot of testing, years and years to find these areas. What they come out with in the end is there are specific regions on certain chromosomes that they know statistically they're able to use and show a unique DNA profile. What is this? If I were to take one sample of your cells at a certain time in the cell cycle, I could isolate the DNA and put it on a slide and look at it under a microscope. Obviously, the right kind of staining, the right kind of microscope, you could pair your DNA up. So your DNA is made of the chromosomes, pairs 1 through 22, and then this is from a female, a male would be X and Y. The regions of interest that we test to build a DNA profile are coming from these chromosome pairs, 1 through 22. On them, there are regions. For example, there might be a band here, and that is something that will be found by the analysis kit that will be tested to make a lot of it that we can actually see it and analyze it. Not every chromosome has something like that. So again, while those researchers are looking at all that, they're testing, are there regions on chromosome 1 that are going to be statistically important for this? Maybe, maybe not. So these are your pairs of chromosomes. One is from mom, the other one is from dad. One is from mom, the other one is from dad, and it continues that way. So when you saw, and we'll look at it again, the blue peaks, there were 2 of them at a specific region. Because what we're doing is we're measuring the length of something on one and the same area on the corresponding chromosome, the length of that one. It'll make more sense when we get there in just a bit. But we went through a lot of testing. They went through a lot of testing to see what part of your DNA works. What we call those regions of interest are short tandem repeats, or STRs. And so again, here's your chromosome 1. They're only showing 1 of them. The previous image had 2 of each one of these. What are these labels? They're the region, the specific area on that chromosome that the reagents detect. We pick up that data. We look at how long it is. So for Elizabeth, she may have a certain region here that is a certain length. She's inherited from her mom, her dad. For me, on chromosome 1 at that region, mine might be a lot shorter. I've inherited it from mom or dad. And we go through that process with all of these regions. So what these are called now are the CODIS, or the FBI regions that are tested to build your profile. Similar to a unique profile to you called a DNA fingerprint. But they're known areas. They're very specific. Anybody doing a forensic analysis trying to get a DNA profile has to use the same type of reagents, has to test the same areas. So we go back to those researchers that had 300 members of a population, and they tested this. They looked at how frequent these are. Does Elizabeth have the same as me? Do I have the same as everyone? If so, then that's not discriminatory. That's not going to help identify me and against someone else and say that person might be a suspect. Again, I'm wording things probably not exactly the way they're worded in a legal situation. I just want to say that. But these regions are reliable, they're inheritable, they're known, they're well tested, and scientists know how frequent this certain information is. So ultimately, what do you have to do? You'd have to be able to run statistical analysis. I'm not going to go into that part. I don't think you'll need it. We'll just tackle the rest of these. So these, let's look closely though. Let's pick one. Chromosome 18. The location says D18. All that means is there's a segment of DNA that contains these little short repeats that we want to look at. So on this chromosome 18, that's the repeat. They're called short tandem repeats because they're short. There's only 4 bases. They're tandem, they get butted up against each other on the chromosome, and they're repeated. I'm not going to go into a lot of these are other examples, but it makes much more sense if I advance to the next slide. So what we're looking at now is an actual section of a sequence of a chromosome 18. And so all of this, if you did a DNA sequence, this is just a portion of it. Chromosome 18 is a lot longer, but we're just zooming in on that little region where the repeat was. So we said on the previous slide, the repeat is AAGA. So the reagents in the kit will detect the repeated regions. So on Elizabeth's chromosome 18, the repeat region is in the same place as my repeat region would be on chromosome 18. But what if she had this length? And if you counted them up, it's going to be 14. They're AAGA, AAGA, you get the idea, and they're butted up against each other. What I mean is there isn't one here and then one over there. Tandem repeats. So this is the part that's unique on chromosome 18 to that individual. We've got 2 chromosome 18s, one from mom, one from dad. Your other chromosome might have a 14, it might have an 18, it might have a 12. There are different lengths. So if you look at enough of these regions and gather this information, you come up with your unique DNA profile. I'm not going to reverse and go back to the other slide, but if you wanted to take time to look at that, go back to the layout with all those regions now, right? The more you test and you're considering how many other people have this in this population is really what you're asking. You realize that you're building a very unique profile, and the probability of someone else having it is probably going to be 1 in however many billion, and probably more statistically than there are actual humans on the face of the earth. And so one example of a short tandem repeat, you'd have many of them tested, and you would use that to build your profile. How do you find these things, right? How do you pick that needle out of a haystack? Because that's really what it is. Your DNA is very small. You are trying to find rare areas, but you have to make sure you're following those FBI quality control standards that you can sit, and show someone, yes, this is because you're talking about paternity, you're talking about solving a crime. You have to make sure you're following all of those rules. So very briefly, we just take cells, whether they're going to be a blood sample or semen or a tissue sample, or if a CSI went to a crime scene and there was a bed sheet there, based on the description of the crime, maybe they would have to take a cutting of that material, whatever it was. So that's why I have biological material here. You put it in a certain reagent, you want to get the DNA out of the cells, the human cells that are there. The reagents we use are specific for detecting human DNA. It's not going to pick up cat, dog, plant, whatever. So we extract the DNA using a series of solvents and reagents that pull away all of the other stuff. Proteins, carbohydrates, things you don't want. They're normal in your cell, but you don't want them in your DNA. Because in the end, you want your pure DNA there so that those kit reagents can literally find and detect those tiny little regions that we're talking about to build your profile. So here you have whatever sample, and yes, these are specialized based on if it's blood or semen or hair, probably assume that. But you'll get at the end a certain amount of your DNA. You have to also quantify it or do a quantitation to see how much you have. Because just like a recipe, you're making a cake, you can't just throw in the flour, you have to make sure you put enough to make sure everything's right. So after extraction though, going back, where do we find those short tandem repeats like we saw on the chromosome 18? So that information, I don't know if you're familiar with polymerase chain reaction. It's a DNA analysis technique where you have synthesized known regions of DNA sequences called primers, and you added a fluorescent tag to those. Those have been built specifically, so there would be a pair of those little fragments that would specifically find chromosome 18, latch on to that repeat area where chromosome 18 area region of interest is, and make a lot of copies of it. Enough copies that you can more easily detect it. That happens at all of those different areas. So the reason they're saying multiplex is you will have different sets of these little primers. One set will find the region called TPOX. A different set will find and copy the D18 and so forth. And there are about 20 of those. And so it's pretty amazing that in a tube about this big and a volume about that much, all of these reactions are going on, detecting 20 different regions of your DNA reliably enough so that we can build your DNA profile. So I realize in a lot of words, and the danger is you trying to read all that while I'm talking, but this idea of those little fragments, finding those regions, they make millions of copies of each one of those regions. So it's pretty amazing that this technology works, is reliable, reproducible, and so forth. So after you extract your DNA and you've proven it's clean enough and you have enough of it, you go through this process of amplification, meaning a specific kit with very specific reagents finds those unique STR markers and regions and makes millions of copies of them. Okay, cool. We got millions of copies. Still can't see it with your naked eye. What are you going to do? More technology using a genetic analyzer. The idea is you can tell that a certain region by the length of it, but you have to be able to distinguish that from that, from that, from that. So when it says labeled with a fluorescent dye, that actual dye color helps. So the way that these are identified after they're amplified would be by the color tag and the length. And so the instrument detects a couple of things like the length of those unique to you and the color tag from the process, and puts the information in bins or says, okay, this is the region we've amplified from D18. Elizabeth has 1418. It labels it. So that's where the numbers come from on the peaks. We'll go through and summarize in a bit. So this is, again, the danger of oversimplifying something, but I think you know where I'm going with that. The way that these areas are detected is reproducible and detectable by capillary electrophoresis. So the analyzer is going to be the magic machine, and that's what I joke with my students. We put the tube in, and it is so user-friendly for students. Hit the button and watch it go, and it literally does everything. They have made these instruments very easy for teaching purposes. So it's called the Seek Studio Genetic Analyzer. Capillary electrophoresis just means that in this Seek Studio, where you lift this lid, it's about this big, and the instrument and the software is about 80,000. So we say thank you to Dr. Gutman for writing an internal grant for that, because he's expanded educational opportunities for anyone who wants to use it. But inside here are these capillaries. So if we kind of took this apart and zoomed in, we'd find these are called capillaries. Very thin, like hair, hollow tubes where the DNA is going to travel through. So we got to get those millions of copies out of that little tube you have from your reactions. And we've got to pass some through this instrument so the instrument can say, okay, that's the region on D18 and it's this long. Or that's the region CS1PO from chromosome 1, if I remember correctly. I may have said that wrong. Forgive me if I did. Right? Identify that. Goes through that for all of them. But the DNA would be pulled if you had your little tube that you amplified. It goes up through the caps. It's going to pass through this area that's going to be hit by a laser. That laser is going to cause those dyes to fluoresce. They get all excited, and there's your red, your blue, your green. So with the length and the tag, those pieces of your DNA unique to you are identified. This is a longer version, and I tend to get ahead of myself. Sure, it drives my students crazy when I do that. But the samples you've copied, millions of copies, they go through the cap. They pass the laser, they get all excited. And this is where I should just put the word magic because it's a long conversation, but the instrument detects, sends the data to the software with all the algorithms, and here are your peaks. The image that I showed just had blue peaks on there. But in reality, when you look at this, you have the different colored peaks simply because they're color coded to the different regions of your DNA. And so whatever this is, it's going to be a unique DNA profile. So you'll have all the regions, peaks there, and the software will give those numbers. The software will put that 14, meaning 14 repeats, like we saw on the chromosome 18, or an 11. That means an 11 repeats, whatever they are. So that's a long way around saying a DNA profile. Now here we don't have those numbers, but you would expect that. So this is from, I tried, actually, I have some more references at the end. So if I did not tag the correct thing, it's the references are there for it. But this is for something I got off the internet, just an example. I just typed in comparison of a DNA profile for a criminal case. Popped up, it's clean enough for an example. A reference would be a person. It might be a victim, it might be someone on the suspect list, whatever it is. And again, there are very careful steps to go through this. You can't just say, ooh, here's the suspect. There's a match, he did it. No. There are very, very specific procedures to follow, but in the end, if someone like me, or you can be looking at it, you can line things up. Now I'm looking at it from the side, but if I remember correctly, that it matches pretty well. What information would be on there that's not would be those numbers like 11 and 12, 13 and 14, whatever they are. And so those numbers would identify the length. But at any rate, a real world situation would be we're comparing an individual's DNA to a sample taken from the crime scene. And so what you're supposed to do and what they do is the evidence from the crime scene, they will isolate the DNA, run this procedure, get the DNA profile. And they can then say, okay, yes, we have enough information to try and make a comparison with other and see if this person, what's the probability that that person left their DNA there? So you don't say this person did it, you run statistics on that. But that's what a DNA profile would look like. So as far as interesting cases, I'm not going to go through lots and lots of details. And again, this and my reference is missing. If what I need to do is make sure, I mean, I certainly want to credit where I got everything, but I will have to give you the better version or the other version. I think what happened was I opened the first one. The other one was updated before I started talking. I actually pasted in. I just want to make sure people know I'm giving credit where credit is due. At any rate, there are old cases. You've heard the term cold cases. So historically, STRs weren't used until after 1983 or so. And even, we all think of, the OJ trial. I'm not going to go through that because of all the problems with that. But one of those problems is when you have brand new technology and you have difficulties explaining it to a jury. Something's new, we just started and we don't have a history of it yet. That's one of the problems. There were others there. But moving forward, now we know these kits, this analysis works very well, it's reliable, and will hold up in court. If we think about evidence that's been taken and packaged correctly, if it's dried correctly, packaged and stored, you can go back 20, 30 years. Maybe you'll have something to test. One of those things that surprised me so much was cigarette butts. So not many people smoke now, or I was gonna say, let's hope they don't. I don't want to offend anyone. But the paper on the part that you put in your mouth would pull off your skin cells. And if it's not exposed to rain and if they picked it up and stored it, that will sit there for a long time. And so that's what was happening here. So apparently there was other evidence, but they could not link the perpetrator or the suspect directly. There were a lot of other details about the case. So again, if you're interested, I'll make sure we have the correct link given to you. But apparently they had gone back to evidence, and one was a cigarette butt. So they took a cutting of that paper that's around the filter. Lo and behold, there we go. Someone who was probably a suspect. But lots of other examples for cold cases. This is something that I'm just now looking at and learning myself. And so we hear, this forensic genealogy. We're having trouble finding enough evidence for a particular suspect. Maybe we have a partial DNA profile or we have something else. Can we go to ancestrydna.com or whatever it's called? And obviously there are legal issues and all of that, and I did not look into that. But let's just say that, yes, here's an open source like this one. And so this says an open source genealogical website. So for this particular paper, that's the Golden State Killer case. So it's not like you can just pull up, grandma's DNA profile or whatever it is. That's not really what's stored in there. You may have a lot of other people, the family tree, the genealogy, and so that has got to be extremely complex. And they're looking at different types of DNA. They're not necessarily looking at STRs. You can't really read it from where you're sitting, but if you looked closely, they may talk about other DNA regions. So in order to use this genealogy, you have open source genealogical information, but maybe you also have a way to look at the DNA in different ways other than what I described and put all that together. That was very complex, but fairly recent. And I'm going to end on something that sounds a little crazy. So when we talk about following all of the FBI quality control standards, that is everything from how you handle and collect the sample at a crime scene, all the way to how the report is written and so forth, and you wrap things up. How do they collect certain samples at a crime scene? I'm not a CSI, but I kind of read this and looked into it. It's very interesting. They use swabs a lot of the time. If there was blood, they'd use a swab, package it, and so forth. In this situation, they were finding a female profile that was not in a criminal database. Oh, is this a serial killer? And her profile was popping up, popping up, popping up. After a while, they figured out, wait a minute, this is somebody who works the line to make the swabs. So I summarized that, I oversimplified that, but this is all about DNA contamination. Going back to the idea of we have got to follow those strict quality control standards. And so sometimes if you look that up, you'll see FBI, or this is ISO, international standards and so forth. But this is really about, heads up, we have to make sure that the swabs are not contaminated to start with. Because here somebody in the very beginning thought, oh my gosh, female serial killer. But if you read this particular female profile linked to 40 crimes, and it took a while to elucidate what that was. So that's all I have. I know I probably got faster than you're used to, or kind of went through some more difficult stuff, but I hope you found something interesting. Yeah.
Y'all have any questions? Yeah.
I do. So for the capillary electrophoresis, I'm not sure if you know this, but do you know if the excitement that happens whenever it passes through is from like the actual bonds from the amino acids, or is it just from the staining of the fluorescent?
So the dyes that are on the DNA fragments called the primers will fluoresce when they're hit with the argon laser.
Okay.
And so those ring structures in that fluorescent dye, and they have little names for them, Ted, Fam, Bill, or I mean, they're just short names. But you can imagine, yeah, ring structures, and they're going to fluoresce when the argon energy, the laser goes through it, because they have to pass that little window. And that tiny little window, and then you see that window here on the cap. That's where the laser would come from the instrument and go through this little window and hit just that section where they fluoresce. It's fun to play with.
Is there a significance to the naming of some of them? Does it include the D? Like, I know that there was a lot that have the D in them, but then there was a handful that don't include numbers or just have like.
Great question. Some of these things, let me back up and say, they're not in a region of your DNA that codes for something important. They're not in a region that makes your DNA polymerase or your enzymes to break down sugars or whatever. I don't know why they name them like that. I mean, if it's 21 on 21, that's pretty obvious. But what is FGA? I'm sure it stands for something and I knew it at one time, but I'll be honest, I don't know now. But CSF, 1PO, TPOX, BWA. When we start talking about a lot of different regions in the DNA or different genes, even just coding for things, some people get a little cocky and name them after themselves, so I. Yeah.
Another one. So whenever you're dealing with, the short tandem repeats or whatever, is it also common to use telomeres in like a similar way? Because I know that it's been like a increasing topic that like telomeres, whenever your chromosomes split, they have this segment that like is repeating and it wears down over time. So like can that also be
After your DNA is copied, like say you have a cell and you need to make 2 cells, you've got to get your DNA replicated and copied so that each cell has what it needs. When that happens through the process, at the very end of the process, part of the DNA is susceptible to breakage. That's what she's talking about. And so if you don't protect that part of the DNA, it'll break off and your DNA is shorter. And the next round of replication, more cells, your DNA is shorter. Eventually that happens naturally, shortening and breakage with age. So there are kits, or there were, they're hard to find now, telomere. So how healthy are you? What is your telomere age? I think Christy Brinkley was one of the ones on the commercial for that, actually, the model. Trying to get people to spend their money, give a sample, and they would say, oh, your telomeres are this long, you're pretty healthy. Talk about misinformation and useless information. But when we talk about repeated regions and things that are reliable, that's what they're going to use. They can test telomeres for other things. There are other repeat regions that they don't use either.
I just wasn't sure if it could be used to kind of like guesstimate around the age of the person, or would that also be something that goes?
That's a great question. I don't think so. I think that's such a gray area.
Yeah.
Yeah. There is a gender identification marker that the kit tests, so they do know X and Y.
Yeah.
That's right.
Yeah. So I watched a lot of Forensics Files and I love that show.
As long as you're not basing your stuff on CSI. I'm
You
kidding.
know, that's my question. Is like I never watched CSI because it's like dramatized, and I'm more interested in like realistic forensics stories that they can tell. I'm just curious what your opinion is on those types of shows, because they are bringing in scientists, but of course it's for entertainment and they're wanting to not necessarily sensationalize things, but make it
I
interesting.
think for the interest of the show, and we were talking about this earlier, was that, they're in the lab and they have a sample in this little tube and they take the little tube and they put it in this instrument, and then I don't know, 20 minutes later, some dude's picture pops up and you're like, oh, we knew it was him. I think the complexity, but it's for the TV, like you said.
Yeah.
But I also, it's my understanding, I mean, I did this for almost 7 years or so, and my employer and other people could certainly speak better to that based on experience. But misconceptions on how long it takes to get something or how easy it is.
Mm-hmm.
I mean, those cigarette butts, but other stuff would be like, I thought this would be a great and for some reason, there might be something in it that makes the reagents not work correctly, and so you don't have a profile.
Mhm.
So there's a lot of things, or you'll have not one person there, you'll have five or something, and so I think that's the problem.
Mhm.
But it's for the idea, but early on, they called it the CSI effect because people were starting to watch those shows and think, oh, there's gonna be DNA, and so maybe some people went, I don't know how prevalent, but maybe some people would show up on a jury and it caused problems.
Right. As a follow-up to that, in more recent episodes that they've been putting out, they've been focusing a lot on the genetic genealogy, and
Yeah.
you touched on it a little bit, and I just think it's interesting that especially for cold cases, they're really starting to turn to that since the Golden State Killer.
And that is something I would like to learn more about myself, because all those other steps, but at least looking at, I mean, how far back do you have to go? You really gotta go, hopefully have that information or some information all the way back several generations. Yeah, so I don't know enough about it to talk about it, but I'm gonna look into it.
If there were multiple like DNAs mixed together
Yeah.
in like a specific area, and you know, you take it and you amplify it, how would you be able to distinguish between the two?
If I write over there on that board, is that okay or no?
Yeah.
I don't know if anybody would be able to see it. Sorry, guys, if I'm out of your way. So again, if you had those peaks, and I'm left-handed, so you know it's gonna be soft. Two peaks like that, one's supposed to be a peak. So if it's one person, the max number of peaks you're gonna have at one region is gonna be two. There's biparental inheritance. Mom, like that. But if you saw this at one area, you'd be like, ooh, there might be two or more people there, because there could be four people. This person could have a 14-14, which means it's only gonna show in one. So all of that with the stats and the identification, all that gets very complex. And I talked, when FDLE comes, and if you are interested in things like this, I don't know how easy it is to actually get a tour of their lab, but they do that for the public. So get 10 of your friends and call them up and say, the reason I say 10 is I think they want bigger groups. And they'll take you through the lab, and they usually come once a year to our bio seminar and give fabulous talk and talk about all this again, and some interesting cases, especially Florida related cases that were solved, and they will sit with you afterwards. They sit with some of the students and just talk for 45 minutes with them. If you have questions, they're awesome. Cool. So
You
Back
said
to-
FDLE,
FDLE, Florida Department of Law Enforcement.
maybe we should do something through honors or with like career development, because they do those career field trips.
Yes, and they do, we do UWF has a good relationship with internships and shadowing with them. They are, and they will tell you they love UWF students. They themselves are UWF students, people that come talk.
That's cool, thank you.
That's great.
Not sure if we have any questions from Zoom. I can see. But otherwise,
It's always touchy trying to figure out how much details go into for certain things, and you don't want to lose anybody, you know, it's like when people start talking about engineering, you're like, I go, I'm sorry, I'm out.
well, we appreciate you so much. There was no questions on Zoom unless anybody wants to. But I'll keep an eye on that for a second.
But I will give you, and since we were in here earlier and I'm trying to find the correct one, the one that I have has more of the links. So that somebody can just click if they want more information, but is that okay if I just email that to you
That's
directly?
totally fine.
And I apologize for making that mistake.
That's totally fine.
I'm
I'm really
excited.
excited.
I'm glad, and it's a great topic. You guys put together some really, really interesting things.
Yeah,
So,
we're so grateful to have you come and speak to us. This was wonderful.
Oh, anytime. It's fun. It's fun. And I can, if anybody's still listening, I do teach the forensic biology classes in the spring. There's still room. We're gonna do some fun stuff.
Definitely.
All right.
Thank you so much for
So
your
I'm gonna
time.
take this out of here, and I think I need to log out. Whoops, I did not mean to do that.
That's totally fine.
Sorry.
You're good. You're good.
Leave it to me to mess up technology. My daughter never watches this, she'll be like.
Totally fine. You can right back up. So I think we're good, but thank you all so much. I'm gonna go ahead and stop recording and we should be good for the day. Thank you all again so much and thank you.